Chamo agora, então, a professora Maria Arménia Carrondo para falar-nos da cristalografia na biologia.
Boa tarde a todos. Eu gostava de começar por agradecer o convite que foi feito pelo professor Dario Moreira dos Santos e a comissão organizadora a estar aqui presente e a representar a biologia estrutural nesta sessão. Já não está à mesa, mas gostaria de cumprimentar todos os membros da mesa que estiveram na fase inicial. Há outro aspecto que eu vou mudar um bocadinho, talvez até ajude a captar a atenção dos participantes ainda nesta sessão, é que eu vou falar em inglês. Eu peço imensa desculpa, mas há termos que é muito difícil traduzir para português e eu correria o risco, certamente, de usar traduções erradas, sobretudo perante um professor de bioquímica e alunos de bioquímica, que eu não me atrevo, de facto, a fazer. Portanto, eu vou falar em inglês e sobre a macromolecular crystallography em biologia. Para começar, quer dizer, eu iria chamar a atenção, what is structural biology and when did it start? Nowadays, crystallography in biology, the general term is actually structural biology more than, as I mentioned, crystallography in biology. And the landmark of the start of this era was indeed, as it was mentioned already, the model, the establishment of the model of the structure of DNA by Watson and Crickson, and with the experimental data of Rosalind Franklin in 1953. They did not solve the structure of the DNA. This was solved by X-ray crystallography only years later, in 1981. But they, with the crystallographic data provided by Rosalind Franklin, they were able to establish a correct model that was later shown to be correct when the structure was solved. And why was this model so important? I guess everybody that works on molecular biology knows, and biochemistry, that this allows to understand the replication, transcription, and translation of DNA. From this era, everything started. And this gave Watson and Crickson, because Rosalind was not alive then, the Nobel Prize in Physiology and Medicine in 1962. So since then, and mainly now, it is very well established and known that structure is function, meaning that to really understand the function of a protein, you need to know the structure. Without knowing the structure is, I would say, impossible to know in detail how the molecule works in a certain organism. And this, of course, is not a unique observation or affirmation, but it goes forward and backwards because structure gives rise to new studies developing the function, and the function leads to new structure determinations, and so forth. And this is how science develops, and this is very important, but indeed, the structure is the main basis to understand the function. So, what does structural biology provides for the understanding of the function? It provides the description of the macromolecule at an atomic level. So what is really determined is how the molecules are constituted atom by atom, and their 3D architectural shape. And with this knowledge, it is possible to know which are the important parts of the molecule, where are the active centers, how it interacts with other molecules, and so forth. So it indeed allows mechanistic information of how the macromolecules and complexes function in biology. What are the methods nowadays used to obtain structural information is mainly X-ray crystallography, is still the more potent method to use to obtain structure from macromolecules. But NMR is also very much used and is increasingly used nowadays. Cryo-EM is a very, very potent method and is being very strongly developed recently. Mass spectrometry and biosacs also is also a very important method. It was already mentioned that crystallography allowed the winning of many Nobel Prizes, and in particular structural biology was one of the strong points on these achievements. And here I'm not listing all the Nobel Prizes awarded to scientists in structural biology. I'm just pinpointing or selecting some of them, and starting by Carrondo and Perutz, which was already mentioned, by solving the structure of hemoglobin and myoglobin. And then Dorothy Hodgkin, Deisenhofer, Robert Hubert, and Hartmut Mikkel, which determined the structure of the first membrane protein, the photosynthetic reaction center. Later on, Boyer, Walker, and School, which allowed, by their structure determination of the enzyme sodium potassium ATPase, to understand the synthesis of ATP. And more recently, McKinnon with discovering of channels in cell membranes, Kornberg in 2006 with the studies of molecular bases of eukaryotic transcription. And in 2009, I believe everybody knows about this example, is the structure of the ribosome won by Venki Ramakrishnan, Tom Stites, and other journals. And I like this in red because I will take this example a bit further. And more recently, in 2012, Lekowitz and Kobilka with the structure of the G protein and coupled receptors. So indeed, structural biology is a very powerful methodology nowadays, and this is the basis of most of the modern biology and biochemistry. So as I mentioned, I'm going to pick up two examples, and I hope to be able to show you how the structure has allowed to understand the function and vice versa. And I picked one example from the literature, the structure of the ribosomes, and then an example from our own work. Sorry about that, but easier was more at hand to also show you these achievements. With the structure of the ribosomes, I will try to illustrate their structure and correlate with the function as I mentioned. So everybody knows that ribosomes are very large complexes of proteins and RNA. Indeed, the ribosomes are the complex factory where the mRNA is translated and proteins are synthesized. And it is composed of large and small subunits which can dissociate and reunite during the process of their function and of their work. What is very interesting is that ribosomes from all three kingdoms of life function according to very similar principles. And many structures are already known nowadays. They really had a major development since 2009. But many more are known nowadays, including the human ribosomes are being determined, the structure. And what the X-ray structure tells us about the ribosome structure is they provide an immense insight into the general molecular and atomic details of protein synthesis and in every organism on Earth. So we can understand how protein synthesis really occurs, and we couldn't understand that without the structure of the ribosomes, okay? And more important, or not more important, but extremely important nowadays, it is the development of new antibiotics. Based on the structure of the ribosomes, of eukaryotic ribosomes, and many others, there is a very strong investigation towards obtaining new antibiotics, which is very critical, as you know, nowadays. Worth noting is that these structures, when they were first determined, were the largest asymmetric molecules solved by crystallography. So they are really huge molecules. So how is the main function of the ribosomes is translation? And very briefly, translation occurs in 3 steps: initiation, elongation, and termination. In the initiation, it is when the mRNA binds to the small subunit, and the first methionine tRNA, which is the substrate, binds to the P site, And then the large subunit comes and associates and forms a big complex. Then the step which is repeated over and over again for the synthesis of the proteins is the elongation, where when we have the mRNA bound to the ribosome, then it reads the codons on the provided by the mRNA, and then recruits the tRNAs that bring the correct amino acids related to that codon, and comes to the A site, and then there is a peptidyl transfer to the ribosome, to the peptidyl transfer center in the ribosome, and then all the system translocates and moves along one step further on the mRNA. So what happens is the polypeptide grows, the protein is being formed, and the ribosome travels along the mRNA. And this cycle, this process, or this step, is repeated as many times as the number of amino acids present in a protein, until a stop codon appears in the mRNA, and then the process is stopped. Okay, stop codon appears, the polypeptide is released, and there is the recycling process where all the components dissociate to be recycled. During this process, and I, of course, am not going, although you will see that in the film that I will show you, during this process, different protein factors are involved that catalyze various steps. The initiation factors, the elongation factors, the release factors, and the ribosome recycling factors. So there are several proteins that catalyze that kind of push to this kind of action in the ribosome. But first, I would like to show you the structure of two subunits of ribosome 50S and 30S. The structure were determined by one of the Nobel Prize winners, Ada Yonath, and published in 2009. This is from Eubacterium radiodurans ribosome. And you can see here, although it doesn't seem very large, but if you imagine that the size of this, you can imagine by the following. Each part represented here on color are proteins, and the silver part are the RNA. So this is one protein, this is another protein, this is another protein. So normally we represent one protein in a big show, but here you have this huge complex which is formed by the various components, as I mentioned, the RNAs and the proteins. And represented here the sites of binding of the substrates to the RNA, A, P, and E. On the other side, we have the small subunit of the same organism, and here represented as a highlight, we have the substrate represented in a backbone shape, and in green it is represented the part with the anticodon that would bind to the large subunit, matching the code of the mRNA, and in pink the amino acidyl stem that will bind to these sites here on the small subunit and delivers the amino acid to the peptidyl transfer center. As I said, also, the structure of the ribosome provided and pushed for development of many new drugs, because ribosomes are target for about 50% of the antibacterial drugs. So indeed, if you find a way of blocking the activity of the ribosome, you kill that organism, and therefore don't allow it to produce proteins any longer, and you can solve the problem, okay, of the infection in that case. So this is extremely important. And for that purpose, the high-resolution structures of the subunits have provided a new area of possibilities of structure-based drug design, and indeed new and effective drugs in the race against mainly the newly developed method of resistance development is being pursued and achieved. Here is very briefly represented again the large subunit and small subunits, the sites of interaction of various antibiotics with the ribosome subunits. Again, the publication from Ada Yonath. And but now this publication is from 2008, But nowadays there are even more sites of interaction. And the various new sites of interaction can allow the development of new drugs and new actions to stop the activity of the ribosome. So I will now show you a movie illustrating translation. This movie was downloaded from Venki Ramakrishnan website. And important to remember is that all molecules and complexes that are represented in this movie, their three-dimensional shape was determined by structural biology methods. So with this in mind, you can see what was achieved by knowing the structure of these molecules and complexes and so forth. Even dynamics events were also structurally characterized. Okay, here it comes. So this is a copyright. Here you have the mRNA and small subunit of the ribosome. Then the mRNA binds to the small subunit. The initiation factors come here, and you have the first step, the first methyl tRNA represented here. And then the large subunit comes in, forms a complex, and the initiation factors are released. Again, this one here. And you have the elongation step starting, where you have the first amino acid tRNA with elongation factor TU coming in and binding to the A site, as you will see. Now, a hybrid state is formed when this tRNA tries to move into the P site. Okay? And the elongation factor G catalyzing the translocation here on the mRNA. It's very visible scene. Okay? So these are detailed parts of the elongation. And then you will see a 30S open form oscillates between one open and closed form. And then you have these processes I mentioned repeated over and over for all the amino acids that constitute the protein. The polypeptide chain grows, comes out through this pocket, a pocket close to the peptidyl transfer center, and is being formed and folded until a stop codon arrives on the mRNA, and a release factor is linked, and a ribosome recycling factor comes in, catalyzed by EF, elongation factor F, and as I mentioned, all the complex is dissociated and the process restarts. So I hope you enjoyed the movie, which illustrates, as I mentioned, the importance of the knowledge of the structure to understand the function of the ribosome. In Portugal, we have structural biology mainly in more developed places where it has been developed for longer times. Is at ITQB, FCT-UNL, and I at BMC in Porto. But now there is a small group in the Faculty of Medicine Veterinary at the University of Lisbon. And I will now show you very briefly an example from our own study at ITQB, which is an example on the topic of pathogenesis of Kaposi sarcomas herpes virus LANA, the protein LANA from the system. And this is a project that under the Harvard Medical School program that was developed on a multi-theme project by ITQB and that took part on the structural studies. The IMM did the in vivo studies, and the group from the Harvard Medical School did the in vitro studies. So herpes virus family is a very large family of dsDNA viruses, double-stranded DNA viruses. And it's important because it established latency, dormancy in the host cell associated with different malignancies, such as the Kaposi sarcomas associated tumor. During latency, the virus is persists in the nucleus of the host cell as an episome, and only a few genes are expressed. And indeed, the protein which was the target of our project, LANA, the name is here, is one of the proteins that is expressed during this process of latency. So while the virus is dormant, is not developing, and therefore the infection is kind of on a holding situation. But if this is disturbed, then the infection restarts, and so it's very important to understand how this takes place. Okay, here I'm representing only the interactions in the host nucleus during mitosis, and you can see here that the protein LANA, which is represented here in green, is a protein that links the virus genome, represented here as a circle, and the host chromosomes, represented here by this shape. So this protein is very important because it keeps the virus in the host cell, okay? And if we want to understand how this happens, it's important to know the structure. So in a way, it allows the viral DNA to piggyback on chromosomes during mitosis. There are other DNA tumor viruses similar to the Kaposi sarcomas, and like the papillomaviruses, which is very well known by everybody, and they also have similar proteins to the LANA protein. Here is the sequence. I will not go very much onto that, but what we have done was the structure of the C-terminal, represented here, and we didn't, although we are also working on the human LANA, but for the moment we only solved the structure of the C-terminal of the mouse model. The structure of the M-LANA at 2.2 resolutions, this is the structure of the model, and I hope you understand these kind of representations of the protein structures. They are not atom by atom, but by secondary elements, like helices, sheets, and loops. Otherwise, it would be very complicated. The fold is similar to the fold of the EBNA1 structure. They work in vivo as a functional dimer, and the interface between the monomers is very important as it keeps thermostability between common to all these proteins. So what has our structure revealed in terms of the function? We have seen two faces, as I indicated in the previous slide. There are two faces on the structure of this protein that we call ventral and dorsal face. And in the ventral face, if we represent electrostatic surface representation of the ventral face, and for those who are used to these representations, these sites with color blue mean positive charges, with color red mean negative charges, and the white or intermediate means intermediate charges. So we see here two very large positive sites. And these are probably the these two strong positive charges where we located phosphate ions are the possible site for interaction of the DNA with M-LANA. So this is in detail what we have on the interaction of these phosphates, so that you can have an idea, indeed, then when we go to detail, what kind of information we can obtain. So we only solved the structure of LANA, not the complex, but we are able, with the structure of LANA, to build a model with the structure of LANA plus the DNA, and the model is very stable and shows exactly that the DNA interacts with the LANA in the ventral site in the two places that I mentioned before, with these positive charges, which is what would be expected since the DNA has on the outside negative charges. On the other side of the molecule, which we call the dorsal face of M-LANA, we found, to our surprise, and different from the similar proteins, a very large positive patch. And when comparing with the similar protein EBNA, this doesn't have the same pattern at all, completely different. And what is the purpose, or what is the function, or why does this protein has this positive patch? What happened was what I described at the beginning. Then we gave this information to the other groups, for the Harvard Medical School and the IMM groups, and they provided the targeted mutations in the ventral phase and also in the dorsal phase, and they were able by biochemical studies to show that these mutations in the places, in the residues that were shown to form these positive patches, were able in one side to eliminate DNA binding, and in the other side to also affect DNA binding and also have implications in latency. So the ventral side, we concluded the ventral side was the site for DNA binding, and the dorsal phase was the site for the association with the chromatin via a complex which is represented here by one of its molecules. So indeed, our study and our structure had impact on viral latency and understanding viral latency. So the very interesting aspect of this overall study that is that it combines structural, as I mentioned already, in vitro and cellular studies that with an animal model that can offer a unique opportunity to investigate viral pathogenesis. This work, altogether with these several contributions, was published last year in PLOS Pathogen. And here is the main people involved in that work. As you see, Pedro Simas from IMM, Ken from Harvard, and from ITQB, Colin McRae, Rajesh, and myself, and student Bruno. And I think it's Sarah here as well. And thank you very much for your attention.